Okay, good. All right. So as we head into our final couple weeks of the semester, so this week we have two lectures, so today and Wednesday. And then this Friday, we will have our very last article share. So I know we had an article share last week, but I like finishing up with another article discussion. So after class today, I'll make the submission link for this week's article share assignment available. So this week, we're going to talk about clinical treatment of cancer. So that's today's topic. We're going to talk about clinical trials. What are clinical trials and what are some of the benefits and limitations of the way that we've conventionally been doing drug development and how we do things like testing drugs in clinical trials. So we'll talk about phase 1 and 2 clinical trials. I'll talk more about different types of approaches for making cancer drugs. This Friday, we'll have our article discussion. And then on Monday of next week, we're not going to learn any more new material, but what I thought we would do is, every semester, I'd like to have the opportunity to share with you the nature of my own research and the work that I've done as a cancer researcher. And so what I often do is there are two main projects that I like talking about. I have the class sort of do an online La Lima poll where I tell you what the two topics are, and then the class can decide, right, which topic you want to hear about next Monday. All right, so I will talk about that more this week. And then next week on Wednesday, we will have our last class meeting, which will be a review for our third exam. And then the third exam will take place during the final exam schedule. So Monday, May 10th. Okay. All right, good. So that sort of little bit of sort of orientation was as much for my benefit as yours, right? Because sometimes I lose track of where we're at. But I make it sound like it's all for your benefit. Okay, let's go ahead and talk about clinical treatment and different types of considerations for how we deal with cancer in a treatment setting. All right. So when a patient presents in the clinic to an oncologist, with a tumor or tumors, one of the real fundamental decisions that a clinician has to make is whether to treat or not to treat. This might surprise you because you might think it's obvious. Well, if somebody has a tumor, you have to do something about it, right? Well, not necessarily. So this has to do with how the tumor diagnosis sort of dictates what the best sort of sensible steps are to take. Tumors are typically viewed in three categories. Number one are what we call indolent tumors. So these are tumors of a particular type or a particular presentation that seem to indicate low invasive or low metastatic potential due to maybe like the tissue type or the current sort of appearance of the tumor. And it's quite possible that the tumor may never really show the signs of developing into something that's cancerous. Right? So in that case, it may actually be that tumor can just sort of persist in the patient and not really pose a big health threat, right? Because it may remain of a particular size for the life of the patient. In which case, any kind of aggressive treatment, the side effects might actually be worse than just leaving the tumor alone. On the other hand, number two, you have highly aggressive tumors that by the time the clinician sees it, likely have already metastasized, right? And so these are sort of like the terrible situation of patients presenting with late stage cancer at the time that it's first diagnosed. And that usually comes about when the patient, waits until they actually are suffering from other types of symptoms like pain, discomfort, right? Other types of physiological changes that finally get them to go to see the doctor. And by then, oftentimes, right? The disease has metastasized. The third situation are intermediate grade tumors that, yeah, if you leave it alone, they likely will eventually get to the point where they're aggressive and metastasized, right? But maybe for now, they can be easily removed or simply treated with different types of therapeutic cytotoxic agents with a very good prognosis. Right? So of these, the scenario number 3 poses the greatest choices for the treatment method. Because we're not saying leave it alone. That would be like in case number 1. We're not saying, wow, we have to get in there, right? And no doubt about it, really try to take some drastic measures to treat heavily, right? What we're saying is, number 3 gives many options for what you would like to try. And the key here, right, is this theme that we've been talking about all semester long, is if you understand the biology of the tumor, meaning the etiology, where it came from, what type of profile it presents in terms of things like intracellular signaling pathways, then, right, you really, as a clinician, have the upper hand in being able to choose the best treatment choice for your patient. Let's talk about sort of conventional treatment strategies or therapies, right, and the impact that they've had on cancer mortality. Let's start off with these traditional strategies down below. All right, so surgery, of course, is based on historic observations that small tumors, right, are the basis of forming large tumors. So therefore, if you can resect or remove primary tumors in time, you can prevent metastatic lesions from happening. Right? Radiation therapy. Right? So radiation, giving high doses of radiation to intentionally disrupt the genomes of cells, right? To actually encourage them to die based upon messing up their genomes, right? That is the basis of how radiation works to kill cancer cells, right? Because remember, cancer cells are still living cells, right? Their genome, even though it might be really messed up after years of, chromosomes sticking together or things being deleted, right? That crazy state of the genome is still intact enough in order for the cell to still be able to use it and survive, right? That's one of the things that we oftentimes don't appreciate, right? That a cancer cell's genome can only be messed up to a certain extent. Right? Once it gets so messed up that critical genes are disrupted, then the cancer cell itself can't survive. So radiation treatment, right, can be used to really try to mess up the cancer cell. So radiation used as an adjuvant therapy, or in other words, in addition to things like post-surgical therapies, can be tremendously successful. And then the third is chemotherapy, right? Chemotherapy, which involves compounds or the introduction of molecules into the body, right, that are either natural or synthetic, and then you are expecting these molecules to somehow do something inside the cancer cells, like disrupt a particular pathway. Okay. We'll talk about chemotherapy a little bit more, but it's worth saying that oftentimes different chemotherapeutic agents, the ones that have been historically used for decades, are often discovered in sort of a blind fashion through natural or synthetic like compound screening, right? And oftentimes they're discovered with very little a priori understanding of the mechanism, right? They find out that a compound works to maybe slow the growth of a tumor, and then afterward, right, there's lots of effort put into figuring out, okay, how does it work, right? So if you look at the top here, this is following some trends in different types of cancers that sort of reflect the effectiveness of these traditional strategies for some types of cancer, but also really highlight how inadequate these traditional therapies have been for other types. On the left are examples of certain cancers that we've seen a decline over the last, roughly 100 years or so since they've been followed. So on the y-axis is the death rate per 100,000 people. Right? So you can see this figure over here tops up at 60, right? So 60 out of 100,000. And so things like stomach cancer in men and in women, colorectal cancer in women, uterine cancer, of course, in women, right? These have all gradually declined, especially starting around the 1950s, right? With the advent of good therapies like surgery, radiation, and the discovery of different chemotherapeutic agents, right? But notice on the right is a different set of cancers. Now, keep in mind, the scale on the right goes up to 100, right? So if the scale on the left is 60, in reality, all of the stuff on the left is from here, 60 down below, right? So the actual heights of these lines are a little misleading if you look at panel A versus panel B, right? But what they're showing is that certain types of cancers, like lung and bronchial cancer, right, have tremendously risen over that same amount of time. Interestingly, this has everything to do with cigarette smoking, right? Because in the 1930s and the early 1940s, during the World War II, right, cigarette smoking became a tremendous sort of pastime. Cigarettes were distributed to troops everywhere, and as a result, right, you had lots of people who started smoking. And so the effects of this, right, carry through decades, right? Because remember, it takes time for cancers to form. And so and then starting in maybe like the 70s and 80s, and certainly in the 90s, then it became more this awareness of it's not healthy to smoke, right? And people began to sort of try to stop smoking, right? And that's why you now begin to see the effects of that tapering down. But other types of cancers as well. For example, colorectal cancer in men, the brown line here, you can see, is actually quite stable for many decades, now only starting to taper off in the 90s. And things like breast cancer in women, right, also quite stable. Right? Pancreatic cancer, ovarian cancer. Here's the pink is the lung and bronchial cancer in women as well, following the trends in smoking, right? So clearly traditional strategies, there's lots of room for improvement. Let's look at these. So let's look at some examples of surgery, radiation, and chemotherapy. So in the early 2000s, so it's been, like 20 years now, they had the advent of better and better sort of robotic assisted surgeries. So this particular company that makes this surgical robot, this is the Da Vinci robot, right? This allows, for example, a patient who's undergoing surgery to be worked on by these very, very precise robotic arms that allow very precise incisions and as well very precise manipulations of tissues inside the patient. You can see these arms are actually being controlled by the surgeon who sits behind this interactive sort of VR type of a control panel, right? So in this case, he can see on a microscopic scale, right, what these individual little prosthetic devices are doing. I remember as a graduate student back in the early 2000s at USC at the Norris Cancer Center, where I did my PhD work, this technology was very new, and they actually came and they set up a demo of this. And of course, the thing was they had free pizza, right? So that's why I went. But as I'm eating my pizza, they're showing that these little arms are incredibly good at fine motor skills. So they had like a bunch of Fruit Loops cereal on the table, and they showed that these arms could individually pick up these Fruit Loop cereals and sort of like string together these Fruit Loops and things like that, right? So that was like, almost 20 years ago. So now we have even better technologies. But this has allowed surgeons to be able to get in and do very invasive surgery with very little trauma to the patient, right? The actual incisions in the patient are very small to just allow these sort of probes and prosthetics to go in. What about radiation? So I was just telling you that radiation treatment is all based upon the fact that cancer cells, right, that have messed up genomes, right, they've sort of stabilized at a point where their genomes can only get so messed up because they still need to function. So if you hit them with high doses of radiation and really mess them up, what you're hoping to do is get the cancer cells, to a point where even their genomes can't handle being alive anymore or sustaining the life of the cell. So what we call this radiation-induced mitotic catastrophe. Okay? So by zapping these cancer cells, so these cancer cells have been zapped by radiation. And of course, what do cancer cells do? Cancer cells love proliferating, right? So especially if you're talking about an aggressive cancer here. So the problem is during mitosis, right, when you have division of the cells, the chromosomes have to be well organized, right, in order for division, in order for metaphase and anaphase and telophase to happen properly. So through radiation damage, if you can mess up the chromosomes enough, then mitosis cannot happen properly, right? You end up getting weird complements or amounts of the genome in either cell, and that hopefully causes the daughter cells to just be non-viable, right? So as these cells divide, right, they are an intact cancer cell that might be functioning like okay. You zap it with radiation, the next time it tries to undergo mitosis, right, there's this catastrophic event where both daughter cells basically cannot survive, right? And that's what you're trying to do. Now, of course, if you're talking about a big dose of radiation, right, what about your normal cells and your normal tissues? Yeah, radiation is a scary thing. That's why, hey, when you go to the dentist, right, and then you're having little x-rays in your mouth, I think I told you this before. I mean, you guys have all been through this, right? They put on that heavy lead apron around you to protect as much of your other tissues from radiation exposure as possible, right? And then they're basically then just zapping very carefully, like your jaw, right, to get your x-rays, right? So all of those normal cells in your tissues in your mouth, they're getting zapped by radiation, right? Not this level, right? Because that's the other thing too, right? Is that they're not zapping enough to actually cause mitotic catastrophe to happen, right? But they're zapping it enough so that you can develop the X-ray film, right? That you're awkwardly biting, right, between your teeth, right? You're like, ah, and they're like, no, hold it this way, and you're like, ah, right? Yeah. So, but still, you're going to have X-ray damage to the healthy tissues of your jaw. So you say, well, then how come you don't end up developing, jaw tumors? It's because those are healthy cells. So if they end up having damaged genomes, what do? They undergo apoptosis, right? So, so it's kind of weird to think that after your teeth X-rays at the dentist, you're going to have several 100 million cells in your jaw just undergo apoptosis, but it's okay. You got hundreds of billions of more of them that don't, right? That are not damaged or that can fix the damage, and you're okay. But the fact that you have to wear a lead apron kind of makes you pause and think, right? Yeah. All right. So why are tumor cells more sensitive to induced genomic instability than healthy cells of your body? Again, it's because most of the differentiated cells in your cheeks and jaw, right, they're not really going to be dividing again, right, because they're already fully differentiated. So it's the dividing cells that would really undergo this mitotic sort of catastrophe, right? So that's why the tumor cells, by nature of the fact that they proliferate a lot more, right, this is an opportunity to kind of target that proliferation as an opportunity, right, to mess them up so that they don't continue successfully replicating. But of course, if you can focus the radiation, right, to minimize exposure of healthy tissues to the radiation, I mean, that's really the best thing. So you can either do that by focused beam radiation, where they basically use two lasers, and each laser by itself is low radiation, so that as each laser is passing through your healthy tissues to get to where the tumor is, right, those healthy tissues don't receive a strong enough dose of radiation to really be damaged. But where those beams cross, right, that point where they cross would be a double dose of radiation, right? And so that's what they do. They carefully aim the beams so that they cross or focus on the tumor cells. It's kind of like that old 80s movie Ghostbusters, right? As a kid, I liked Ghostbusters. I don't even know if you guys have seen that, but they're always worried about crossing the beams that they use to like zap the ghosts, right? Okay, I'm not going to go off on that because there's really no relevance to that. All right. Another way you can focus radiation is by actually, instead of beaming the radiation in, you can have radioactive materials put into the tissue. So that's what brachytherapy is, what we call the seed radiation therapy. So these are little pellets of some type of metal, right, that is a radioactive isotope of the metal. And each one of these little pellets doesn't give off a lot of radiation, right? But you can surgically implant these. So for example, here is a prostate cancer patient. Okay. And you can see in this X-ray, they've planted numerous of these little seeds in here, right? Each one of these seeds is radioactive, but because there's a whole bunch of them focused in this area of tissue will receive a lethal dose of radiation, right? Whereas the tissue surrounding these seeds, right, doesn't get a high enough dose. So the benefit of this is that the overall dose of what the radiation you're putting into the patient can be lower, because this is not something that has to be done where the patient only has an appointment, and during that appointment you're beaming them with radiation, right? This is something that's surgically done, implanted, and the patient can carry these seed radiation pellets in their tissues for quite some time, days or even a couple weeks, until they come back in, and then they can actually see how the therapy has progressed. Okay. So therefore, when you look at these traditional strategies of surgery and radiation, even though I've mentioned a couple examples of how technology is getting better, where we have these surgical robots that are getting finer and finer in terms of their motor skills, and VR headsets that allow the surgeons to see what's going on inside of the body in 3D. And we talk about radiation, how we have these things like little pellets of radiation that you can intentionally implant in the tissues, right? Those are getting better. But those types of improvements in methodology, right, don't really focus on the fundamental question of biologically, where did this tumor come from, right? Surgery and radiation is all about just trying to get rid of the tumor, just as a physical thing that needs to be kind of destroyed, right? It doesn't really place much emphasis on what is the underlying biology that gave rise to this tumor in the first place. That's why the third choice, chemotherapy, actually has the most room for advancement, right? Because the more we understand about the different complex etiologies or origins of different cancers in different normal cell types in normal tissues going bad, going rogue, and causing disease, right? The more like we have a broad horizon of different options for developing things like new drugs that can target pathways that maybe previously were not targeted before. And the important thing here is you can be very discriminatory, right? Rather than brute force approaches of physically removing tissue in surgery or zapping tissue like with radiation, right? If you can design custom tailored drugs that go after very specific intracellular pathways, right, then you have the very desirable option of just specifically targeting a particular cancer, right, in a way that spares surrounding tissue. So here's some early examples that kind of highlight this, right? So one of the earliest types of chemotherapeutic agents was retinoic acid, right? So retinoic acid is actually an acid normal compound that's produced in the body. Retinoic acid is utilized as a ligand for different types of receptors, right? But one of the things that retinoic acid does is it encourages differentiation of cells, right? So in other words, that's one of the ways that certain cells receive messages to differentiate and become like the full differentiated version of what they're supposed to be. So early on, oncologists were studying the fact that you can expose tumor cells to retinoic acid and you can encourage them to differentiate. So here's an example. Here you have some promyelocytes, right? So these promyelocytes these are not necessarily disease cells. These are just sort of progenitor cells of immune cells. right? So these are sort of stem cells that are going down the path of becoming differentiated cells. But if you give them retinoic acid, you can actually get them to commit to becoming normal immune cells like neutrophils, for example. Right? So you can do this with cancer cells as well. So here's a question. Why would forcing cancer cells to differentiate be a good anti-tumor strategy? Right? You're thinking about, these are cells that are becoming cancer, they're transforming. Why do you even worry about trying to get them to differentiate? Right? It's because, remember, one of the fundamental principles of cell biology that we began this semester talking about is that a given cell at any given time, right, is either focused and busy doing its differentiated cell functions, or it's focusing on dividing, right, and getting to mitosis. So a particular cell cannot be doing both things at once, because the way that the genome, the DNA itself, is accessed and used during normal gene regulation in differentiated cells versus DNA replication, right, are fundamentally different mechanisms. You can't have them happening at the same time. Things would get really screwed up, right? So therefore, if you can encourage cells that have sort of lost their interest in differentiating and are shifting towards uncontrolled proliferation, if you can induce them, right, to differentiate and commit to differentiating, you can potentially reverse some of the transformation that's taking place. Okay. Let's look at some examples of some classical chemotherapeutic agents. Okay. These are some examples here of small molecular drugs that target DNA, right, as the substrate to sort of act as anti-tumor effects. So some of these form DNA adducts, or they cross-link with DNA in an attempt to kind of mess up the structure of the DNA so that the cancer cell cannot properly, utilize the DNA and it cannot properly divide. So many of these are small compounds, like alkylating agents, like these over here. They simply form covalent bonds with DNA. Carboplatin. So a number of you have mentioned platinum-based drugs. So carboplatin and cisplatin are all based around the metal platinum, right? So you can see they're actually very simple molecules. So cisplatin actually is platinum with 2 amine groups, and I think it's 2 chloride groups sticking off of it. If instead you have this ringed structure, this is carboplatin. Right? But still a very simple little structure. Right? And it's an alkylating-like agent in the sense that it forms covalent adducts with DNA. Some of the compounds that are being used are what we call nucleoside analogs. So if you look at these structures, hopefully from your Bio 275 days, you remember, oh yeah, right, these are nucleotides. So you have a nitrogenous base of some sort, right, a ringed sugar group, and then in this case, you even have a phosphate group at the end. Right? So fludarabine here, right, looks very much like one of the nucleotides that you would see in DNA, except, right, it's different. Right? So it's the structure is slightly different, the base is different. That means if you have this stuff floating around inside of a cell that's trying to undergo DNA replication, right, and you have the DNA polymerase trying to copy the template DNA, and it's putting together nucleotides that are floating around, right, A's, T's, C's, and G's, stringing them together. If it grabs this and puts it in there, then the copy strand that you make is screwed up, right? And that's not going to be usable for the cell. And so that way, when the cell tries to go ahead and complete replication, it gets all messed up, right? So nucleoside analogs are a very good way of disrupting DNA replication. Okay. Let's take a closer look at the cisplatin drugs, like the carboplatin, like the cisplatin. Here again, here's the cisplatin, right? So I told you that it's very simple. It's a platinum, 2 amine groups, 2 chlorine atoms. And the carboplatin's a little fancier because it has that sort of ringed structure around it. Right? So here's the little space filling model. The reason I like cisplatin so much is how simple it is. Right? When you look at it, that's all it is. It's platinum with 2 chlorines and 2 amine groups. And yet it is one of the most popular frontline chemotherapeutic agents. So where did this come from? How was it discovered? It was discovered in the late 1800s, and it was called, given this name, Péron's salt. Péron's salt, right? So for many, many years after that, this was just sort of a byproduct of different organic chemistry types of metallurgic reactions and things like that. You can make this cisplatin. However, there was an interesting discovery in the 1960s that led to the use of cisplatin as an anti-cancer drug. And that is that there were some researchers who were actually working on E. coli, all right, and they were running gels. They were running gel electrophoresis, trying to analyze components of, running an electric field through bacteria to see what would happen, right? Their main interest was just seeing how the E. coli responded to electricity. But because they ran it in sort of this like gel-like chamber that had electrodes and I think some buffer around it, what they discovered was, for some reason, the electrodes would be building up these sort of products around them, which had to do with the electricity flowing through these platinum electrodes, right? And you get this compound, and this compound seemed to actually prevent the E. coli from dividing. So this is the paper from 1965, right? Inhibition of cell division in E. coli by electrolysis products from a platinum electrode, right? And so what they inadvertently discovered was cisplatin. So how does cisplatin work? Cisplatin works by forming adducts on DNA, right? So here it is, that very modest, very simple looking platinum, right, with the its chlorine atoms and amine groups. So the amine groups can latch on to the nitrogenous bases of DNA, right? And it causes this adduct. So as a result, it causes the shape of the alpha helix to be deformed. And the thing is, it's very hard to get the platinum off of this because the bond is it's covalent, right? And it's very strong. Right? So the way the cell has to deal with this is, if you have an adduct like this, the cell actually has to excise or cut a segment of the DNA out that has the adduct, and then count on the fact that you would have DNA polymerases come in and fill up the damaged part. Right? But if you have a lot of these adducts formed by the platinum, right, then maybe the cell can't deal with it. And that's when P53, instead of simply pausing the cell cycle and allowing the repair to happen, if the repair is not successful because you're constantly having more of these platinum adducts form, then the cell will probably undergo apoptosis, right? And if it doesn't, well, it's not going to be able to properly divide anyhow. Right? So the beauty of platinum-based drugs like this is because it's so small. Because it's so small, it just floats right through tissues and floats right through the cell membrane, right? This is not a big complex hydrophobic molecule. It's not a protein, right? It's a tiny little platinum ion with a couple chlorines and a couple amine groups, right? It can float right into the nucleus and bind to DNA. Right? So cisplatin and the more elaborate carboplatin form, they have similar efficacies in terms of working very well, but carboplatin tends to be tolerated better by patients. Okay. So my interest in cisplatinum is that when I did ovarian cancer work, I was interested in understanding how it is that patients who initially respond very well to cisplatin and carboplatin, these are ovarian cancer patients, why in some cases, right, their tumors would actually develop resistance to these platinum-based drugs. And to me, I found that very curious, because again, if these drugs, these simple molecules can just flow right into your cells and bind to your DNA, it's almost hard to imagine how a cancer cell could learn, right, to resist that. So I can talk about that when I talk about my own research, if that's a topic that ends up being chosen. Okay. Here are examples of more complicated compounds that are useful for targeting proteins within the cell. So you can see these are pretty elaborate, right? These are like crazy organic chem structures here. But etoposide are inhibitors of topoisomerase. So remember, you have this enzyme topoisomerase that's critical in actually the unwinding of DNA during cell replication or during DNA replication. So if you prevent the protein from actually unraveling DNA, then something like a cancer cell cannot divide, right? Bortezomib is a proteasome inhibitor. The proteasome is that critical recycling barrel in the cell that's constantly recycling old proteins, breaking them back down again, right? So without the proteasome working, even a cancer cell cannot continue surviving because it has no way of recycling its proteins, right? So bortezomib is very effective as that. Then you have these two examples down here. You have taxanes on the left, like paclitaxel, and you have vincristine on the right. These work on microtubules, and these I like because they are natural products. Okay, but look at these structures; they're horrible structures. Can you imagine, like, if you have like flashbacks to your Chem 272 or OChem tests where you're given like something on the upper part of the page and something on the bottom, and you're supposed to draw out how you can derive the different steps to get from this to this, right? This would be like your worst nightmare, right? To somehow synthetically derive one of these structures, right? But let's look at these and see why these are so interesting. Let's start off with the taxols. So both of these, taxols and vincristine, I said they work on the microtubules. As a quick reminder, remember the microtubules are those protein fiber structures that make the spindle fibers. The spindle fibers that are really critical for attaching to the condensed chromatid pairs, right, and pulling them apart during anaphase. Right? So that involves these microtubules dynamically growing and then being sort of reabsorbed or contracting in order for this to happen. So, hey, even a messed up cancer cell relies upon microtubule spindle fibers as part of replication, right? So if you can disrupt these microtubules and prevent mitosis from happening, that sounds like a very effective anti-tumor agent. Right? So recall that these microtubules are made of these alpha tubulin and beta tubulin monomers. They form this cylindrical tube. So taxanes, they bind to beta tubulin, all right, and prevent beta tubulin from functioning properly. Vincristines bind to alpha tubulin, right, and prevent alpha tubulin from functioning properly. So in the case of beta tubulins are essential in microtubules extending, right, because microtubules extend towards the positive end, the beta tubulin end. Alpha tubulins are important for microtubules contracting because microtubules are broken down from the minus end, right? So that if the other end is stuck to a sister chromatid, as this end is being broken down, that's how the microtubule retracts. So you can already see these two different drugs work in different ways on microtubules. Paclitaxel is a fascinating story, right? So paclitaxel is one of these success stories from these big natural compound drug screens. So in the late 60s, there was a lot of money at the NIH to do these natural compound screens, because back then they were all about, let's get out there, let's try to find these naturally derived products from natural sources and find out if any of them could provide the basis for being anti-cancer drugs. So from one of these screens, they isolated this particular compound, paclitaxel, from the bark of this particular tree. This is the Pacific yew tree, Taxus brevifolia, right? So that's where the name Taxol comes from for this drug, right? And again, wrote a remarkable structure, right? This is something that, you know, no person would have intentionally made this unless you had a reason to make it, right? But Mother Nature in this particular tree was making this. So it was discovered in 1971 from this big NCI-funded plant screening initiative. And it was shown in 1978 to have anti-leukemia effects, right? So back then, boy, you could go out, collect thousands and thousands of compounds from different things, and then the NIH would give you money in the lab to go ahead and just try experiments where you treat cancer cells, right, with some of these extracts and see what happens, right? These days, unfortunately, those types of fishing expeditions are no longer popular, because I think these days they're sort of thought of as just throwing a ton of money at very little, you know, potential for return. So the problem was by the early 80s, you know, they were processing the bark from the specific yew tree, but they had a problem, right? They estimated that about 60,000 pounds of bark would be needed to meet the demand for this anti-cancer drug, right? And unfortunately, wouldn't you know it, the Pacific yew tree is actually, I think, an endangered species, right? So there was this big race to try to synthetically come up with a synthesis pathway to make paclitaxel in the lab, purely synthetically, right? This is where your whole OChem flashback nightmare comes from, right? And so Bristol Myers Squibb, they won the contract because they came up with a way to make the synthetic production, and they copyrighted this, right? So the drug is now, ever since the early 90s, called Taxol. Right? So it binds to beta tubulin and prevents the depolymerization of the spindle fibers, right? So you might say, well, that's kind of weird, right? Because it's a natural compound and they learned how to make the natural compound in the lab. And yet now they're able to like copyright it, right? And make billions of dollars off of selling it, right? Something about that kind of seems to rub you the wrong way, right? But that's the way, you know, drug manufacturing works. So, but there's still hope for improvement though. This is Abraxane. So the compound Taxol, that big crazy molecule, when it gets into your system, you know, a lot of it gets broken down, right? A lot of it just gets removed from your body in urine. So in the early 2000s, this company over here, right, Celgene, came up with a way to actually take paclitaxel and go ahead and combine it with albumin proteins. So albumin proteins are the proteins in your blood that help carry around small compounds so that you don't just end up eliminating these compounds, you know, in your urine. Right? So they came up with a particular formulation of albumin carrying paclitaxel. And because this is a new formulation, because technically speaking, this is a new chemical form, right? You have an albumin protein with paclitaxel on purpose put on it, right? As a result, you can copyright this again, right? This is where things get really crazy because it is a new chemical structure, right? So Abraxane was approved for the treatment of lung cancer, breast cancer, prostate cancer. And as of last year, 2020, right, the sales of Abraxane are over $1 billion per year. Right? Really makes you think about this whole nature of big pharma, right, and how they approach making new compounds and being able to trademark them. Okay, so vincristine and vinblastine are two other examples. So vincristine has been known for many, many generations as a folk remedy for diabetes, right? The plant is highly toxic, right? But that one of the toxic components is this vincristine. And it's toxic because, right, it disrupts microtubule formation or microtubule management in the cells. Right? So in the 60s, this component, the vincristine, right, was isolated, and it's been marketed as Oncovin. Right? Okay, I like these stories, the Pacific yew tree and this vincristine, because I like showing you the pictures of them, right? It's wonderful to think that these trees with the little red berries or these little purple flower here, right, the rosy periwinkle, right, that are just part of Mother Nature doing their own thing. They produce these compounds for who knows what reason. You know, it might be that the vincristine actually is produced on purpose by the rosy periwinkle as a toxin so that deer don't eat them, right? But it's amazing to think that we can find these compounds in them and we can use them for their anti-tumor effects. And it does make you wonder, right, what is still out there that's ready to be discovered and isolated. Okay, so to wrap up then, those were all examples of compounds that were sort of more or less discovered without any real rational notion of specifically what you're going after, right? Especially stories like those big NCI-funded natural product screens that led to, oh, if you have the bark from this tree, there's a compound in the bark of this tree that seems to stop cells from growing in the lab. Or, oh, there's this compound from this pretty little purple flower, right, that stops cells from growing in the lab, right? But those are all discovered without any real rational kind of target, right? So rational drug discovery then is really where the exciting stuff comes to play in terms of what you can do if you have particular pathways in cells that you want to target. Rational means, right, you're thinking something out, heading towards a very particular target that you're dealing with. So this is a whole area of identifying or inventing novel therapeutic compounds based on biological understanding of what your target molecules are. So the 4 considerations, and I'll present these to you today, and we'll stop and continue with this on Wednesday. These are the things you have to consider, right? If you want to be involved in de novo drug discovery, right? De novo means brand new, something that starts from scratch, right? Not taking something that pre-exists and just modifying it. One is you have to consider the feasibility of your target. For example, is your target in the cell a receptor? Is it an enzyme, right? Because you have to be able to know how those things work. What is the target space of that molecule? Meaning it's a 3-dimensional structure. Right? What is the actual space on it that you're trying to target with another compound? Is it a ligand site? Is it a catalytic site? Right? If it's an enzyme versus a receptor. Number 2, specificity. How do you know that this compound, if it binds successfully to your target molecule, how do you know it's not also going to bind to other molecules, resulting in cross-reactivity? Right? Number 3 is bioavailability. This compound that you've designed, that you've done experiments with, right, in a dish of cells in the lab, once you put it in vivo, in living tissues, whether it's an animal or a person, right, what happens to this compound? Is it even going to be bioavailable to the target tissue? Right? Or is it just going to be neutralized by other components in your blood and your urinary tract, right? Like your kidneys or your liver? And 4th is efficacy. In other words, it's one thing, right, to see in a dish of cells how well this compound works and you get very excited, but it's another thing to really see overall, will this really work in a living system, right? Whether you're talking about in vivo animal models or getting to the point of being able to conduct human clinical trials, right? So these are all things that we will talk about in our next lecture as we sort of head towards wrapping up our semester. Okay, all right, I'll go ahead and stop there.